Technical Veterinary Learning, TVL channel. The founder and the producer is the Professor Ahmed Mamdouh El-Sherif, Professor of Infectious Diseases and Epidemiology, the ex-head of Department of Veterinary Medicine, the ex-vice dean of Faculty of Veterinary Medicine, Bani Suef University, Egypt. Ascariasis of the ruminants. Ascariasis of the ruminants characterized by stunted growth and afebrile diarrhea, sometimes with obstructive jaundice, intestinal obstruction, respiratory signs, petechial hemorrhage in the lungs, and fibrotic spots on the liver of the seized animals. Pathogenic risk factor. Number 1, Toxocara vetulorum. Number 2, Ascariasis lumbricoides. The image in this video showing you the female of the Ascariasis and the male. Eggs of Ascariasis species survive in the entire environment for long years. Eggs of Ascariasis species destroyed by number 1, direct sunlight, number 2, ultraviolet rays, number 3, iodine, number 4, prolonged dryness, number 5, high temperature, more than 50 centigrades. This is an image of Ascariasis lumbricoides decorticated egg. This is another picture of Ascariasis lumbricoides decorticated egg, where large ovum appear with thick shell. A picture by endoscope showing Ascariasis lumbricoides inside the intestinal lumen. Adult Ascariasis lumbricoides. Life cycle. Adult worm or L5 put eggs that hatch to first stage of larvae or L1 that molt to second stage of larvae L2. Within 7 days it molted to third stage of larvae or the infective stage that penetrate intestinal wall, reach to portal vein, liver and bloodstream, as well as going from them to bronchi, bronchioles and lungs. The larval stage of Ascariasis developed to adult worm within 8 to 9 weeks post-exposure to infestation. Larval stage of Ascaris reach to intestine within 3 to 4 weeks post-exposure to infestation. Susceptibility. All wild and domesticated ruminants are susceptible to Ascaris infection, as well as young ruminants and adult ruminants. Geographical distribution of Ascariasis. Ascariasis is a worldwide disease. Sources of infestation. Sources of infestation by Ascariasis are infected pasture, infected food staff, and infected water, etc., etc., and etc. Mode of transmission. Number 1. Ingestion of contaminated pasture, food staff, and water. Number 2. Ingestion of infested milk and or colostrum of dams, as mature worms may be found 10 days post-calving and or lambing. Number 3. Intrauterine transmission from infested dams. Pathogenesis of Ascariasis disease. Adult worm or L5 put egg that hatch to first stage of larvae, L1 that molt to second stage of larvae within 7 days, molted into third stage of larvae, which is the infective stage that penetrates the intestinal wall, reach to portal vein, liver, bloodstream, bronchi, bronchioles, and lungs, cause damage in all their passages. Ascaris migration through liver cause hemorrhages and or fibrosis. Ascaris migration through lungs cause edema, consolidation, and or alveolar angiosis. Ascaris worms collection in the intestinal lumen cause obstruction, enteritis, and rupture. Diagnosis of Ascariasis. Number 1. Clinical pictures. Pre-patent period of Ascariasis is 3 to 10 weeks post-exposure to infestation. Most common symptoms are decrease of appetite, stop suckling of dams, stunted growth, rough coat, diarrhea or stop defecation, painful colic, chloroform or the smell of crunchy apples coming from the mouth, dyspnea and paroxysmal cough. This image showing Homunculus contortus infestation that leads to submandibular edema, or what's called in veterinary medicine bottle jaw, as it appears and demonstrated by the yellow arrow in this image. This image showing Homunculus contortus infestation that leads to submandibular edema, or what's called in veterinary medicine bottle jaw, as it appears and demonstrated by the yellow arrow in this image. This image showing Homunculus contortus infestation that leads to submandibular edema, or what's called in veterinary medicine bottle jaw, as it appears and demonstrated by the yellow arrow in this image. This image showing Homunculus contortus infestation that leads to submandibular edema, or what's called in veterinary medicine bottle jaw, as it appears and demonstrated by the yellow arrow in this image. This image showing Homunculus contortus infestation that leads to submandibular edema, or what's called in veterinary medicine bottle jaw, as it appears and demonstrated by the yellow arrow in this image. This image showing Homunculus contortus infestation that leads to submandibular edema, or what's called in veterinary medicine bottle jaw, as it appears and demonstrated by the yellow arrow in this image. Image of goats infested with Ascariasis worms showing pale mucous membrane. A picture of calf severely debilitated due to internal parasites where loss of condition, green scour, edema, and bottle jaw appearance. An image of calf severely debilitated by internal parasites, loss of condition, and green scour are clearly appear. Postmortem examination. A dead lamb due to heavy infestation with Ascariasis. Abomasal lesions due to Startagia larvae. Hyperemic gastritis. Laboratory diagnosis of Ascariasis depending upon collection of samples from suspected animals directly from the rectum or from freshly fecal sample. Send it to the lab within 4 to 7 hours in refrigerator, and then doing of flotation technique to count the number of Ascaris egg per millimeter, or making direct microscopical slide to see the Ascaris egg, as well as seeing of the adult worm in the fecal matter is a highly indicative lab test. Differential diagnosis of Ascariasis will be done from all infectious diseases that causing diarrhea for ruminants, either large or small ruminants. Differential diagnosis of Ascariasis will be done from all infectious diseases that causing diarrhea for ruminants, either large or small ruminants. Treatment of Ascariasis. Ascariasis are treated by parasiticides or anthelmintics. Properties of an ideal anthelmintic or parasiticide. Number 1, broad spectrum of activity. Number 2, non-toxic at the therapeutic dose. Number 3, have a wide range of safety for treated animals and users. Number 4, rapid metabolism and excretion. Number 5, leaving no residues in the milk or meat that going into human foods. Number 6, easily administered. Number 7, cost effective. This table showing active compounds such as triclopyrdazole, nitroxanil, chlorosone, oxyclosanide, which produced by 2 different companies. Also, this table includes the trade name of this active compound, the company that produced them, doses of them, packaging quantities that present in the marketing, and contraindication of each drug for using. This table is very helpful for veterinarians in treatment of broad spectrum of internal parasites, either for large ruminants or small ruminants. This table also showing the active compounds with names of ivomic, oxyclosanide by different companies, albendazole and ebendazole and ricobendazole with their trade names, companies of production, doses per body weight, packaging quantities in the marketing, and contraindication for using of them in different animals. This table is very helpful for veterinarians for doing of strategic dosing and treatment of any ruminant, either large or small, are infected by internal parasites. Prevention and control of Ascariasis. First step is isolation and treatment of affected animals. Number 2, non-specific alteration of pasture. Number 3, avoid grazing post-training and or in moistened pastures. Number 4, strategic dosing with anthelmintics. Number 5, application of biosecurity on barns. Keep on the track for having the most accurate, simplified, and active information about infectious diseases and epidemiology by subscribing this channel.